where we obtained the genes, diseases, mutations if there were any, and chemicals and species from 150 papers. Now, what is the next step? What do we want to do in the next step? So, we understood that each of these is Anthrax ID. The genes that are genes means that are mentioned as per their name or some other alias symbol, they are all mapped to the same ID, which is the Anthrax ID. So it is some kind of normalization. So the same gene is known in different ways, and authors use them in different forms in the literature, and therefore we need some kind of a standardization if we want to do data analysis. And that standardization is offered by PubData by doing the annotation and mapping the different variants of a particular gene terminology to one Anthrax ID. Now, having done that, we need to get some more additional information. Without getting additional information, then also we can probably proceed. So we'll try both ways. So one way is that we can write some small R scripts which can process the data in the table, the Excel table I showed you. So basically what we do is that we can import the data from Excel, and here we can click on browse and give the path of the file. The file is right here. This is how we saved it yesterday, Excel. There is also a text file. We saved it as Excel and click open. And after clicking open, you can already see the preview of the same table in RStudio. This is a data frame. This is a data frame from the same Excel table, which we have got in RStudio. And then now here, there is a button called import. So click on import. And you can see here it has appeared. And this is where the table is with 149 observations of 6 variables. So here are the genes. So in some paper, yeah. So in some papers, there is no mention of any gene, like here. Whereas in other papers, there are mention of genes, a lot of them about miRNAs, because we had selected a subcorpus of the original main corpus. Diabetic retinopathy is the original main corpus. From there, we have created a subcorpus, and that subcorpus is miRNA because it is involved in gene regulation. It is also a hot topic of research because the miRNA expression and the changes in its expression do a lot of things and in the disease process. So this is a very important and hot area of investigation in many diseases, and diabetic retinopathy happens to be one of them. So we can see the disease column here. Then we have the mutations. There are no mutations studies reported. Then chemicals such as glucose, lipids, oxygen, etc. And then we have the species in different ways. It's all human form only. Patients, people, and as you keep going, you can see somewhere there is no species reported here. So here only disease is mentioned, nothing else in this paper. Then there are other species like rat and mice, which are animal model systems often used to investigate the disease process through molecular analysis. Now, what we have to do is that we have to take out these IDs. You can see these ID numbers. From here, we have to take out these ID numbers because the ID numbers are important. The genes are there, but they are mapped to the ID numbers. So the ID numbers are important. So how do we do that? We can write a small R script here like this. So we already imported the Excel file. So the first line is not required. But the second line, we can run it. And how do we do it? Because this result file, which we imported from Excel, this result file is a data frame. So you can use the dollar operator here. Using dollar, you can select the fields, the data fields, or columns in common language. And once you have selected genes, because that's what we are looking at now. Yesterday we saw the disease process a little bit. Now we are looking at genes. And all I have done is I have put my cursor here and then I click here run. And immediately it takes that command and runs it in the console. I don't have to copy, paste or anything. So this is the help provided by RStudio. Now what I have done here in this script, let's understand this a little bit. So if we look at this data, you can see that there are multiple genes in a particular paper and they are comma separated. Okay, so this is the gene symbol separated by a greater than sign with untrace ID, and then it is comma separated like this. The next one happens to be a miRNA gene, and this is its corresponding untrace ID, and that is again comma separated to another gene like that. So each in each case they are comma separated. So the L apply here, what it will do is it will run the function that we give here inside this curly bracket, whatever function we give. it will run that over each and every element of this column. So the result $genes actually is a, because single column is in mathematical parlance, it's called a vector. So this becomes a vector, and you can give this vector here any number, it doesn't matter. And what it will do is it will run this function over the elements of the vector, the individual elements of the vector, and whatever result comes after running it in the on that individual element, that result will be output as a list element. So if there are, let us say, 10 or 20 or 100 elements in this vector, then it will output 100 results, and each individual result will be numbered 1 through 100 in one data object, which is, in this case, I have given the name my gene IDs, and that data object is a list object. So the list is really very useful in R. And what is the function I am running over each element here? I am running a string splitter basically. So the string splitter str split, it will take the value which is present in each element of this vector and bring it here inside the string splitter function, which is now going to split on the basis of comma. So this is something you have to understand the function and then proceed while writing the script. Why it is called a script? Because it is a one line. It is not a function. It's not a generalized function. This result is the file name that I have generated. In a function, there is no such thing as file name. You can give any file. So that's the difference between a function and a script. So this is still a script. And people have different ways of working. Sometimes they realize that if a script is used very frequently and it is very useful, then they will convert it into a function and keep using it. But if a script is used for one-time task, or let us say few-time task, then it will be kept as a script only. And now when you are going to use the script from somebody, it will work, but you have to know that what was the data object name that person has used for that particular data frame. That is very important. So many times in scripts, some comments are written which can explain what they are doing. Then other users can use that script and modify accordingly. If no comment is written, then one spends hours and hours and hours. Finally, they throw it and say, okay, let me do it from the beginning. So scripts are useful, but it comes with experience how to use them. Even I take time in adopting scripts, what other people have written, because I have to understand what is happening. And then I know that the only basic thing is the data object name. If I have given a different name, then I have to incorporate in the script. And many students, they struggle simply because of this type of uh, points they try to not able to identify and spend a lot of time. So I just wanted to spend some time here in order to differentiate between a script and a function. It is better to write function, ideally. That is a good practice, because once you write a function and write the details about it, then other people can use it and follow. Okay, so we have the str split, which will split it, but then the SPR split, what it will do, it will give the result in the form of a list. So we use the unlist function within this to unlist that and finally get this uh, data into this my gene IDs. So let's look at one, how it looks like my gene IDs. My gene IDs. Let's look at the first one. So after it has split from the comma, we can see that it has 7 elements. Now this, what was a single element from result $genes for the first paper, it has now become 7 elements. But it is still kept together in the first element of the list, corresponding first element of the list. So that is why the list is really in R, it's really very helpful. Now, what is the next step? So in the next step, what we do is we want the IDs from here. So we run the next script, which uses the previous data object, and I click on run. Now here what is happening is that you will notice that the lapply function is used 2 times, one within the other. So that's the advantage in R, that you can use a function one within the other, because this is also a set of elements within the first set of individual elements within the first list element, you see. So we need another lapply to run on this, and now the splitter is this greater than sign. And I want the second part of that splitted data, because the second part actually gives me the IDs. So now let us look at this list, my gene IDs 2, and the first one, and there you see we have got it, each individual ID. But this is still not complete, because this ID has been obtained as a character. Now, why it was character? Because in the beginning here it was character, so it kept it as character and it will continue as character here. And str split will work on characters only. It's a string splitter basically. Now we have to convert these IDs into numeric. So we run the next line of the script and say run. Now what has happened? The same my gene IDs, first we unlist it. We unlist everything. Means it completely collapses the whole thing so that we get all the IDs together from all the papers. We can also do it for individual papers, but this time we'll do it from all the papers. And so it says that it has introduced NAs somewhere because somewhere IDs may not be there. So it has introduced NA there. And now we have these IDs, which you can see it is like this. My ID is 2. And let's see the top 5 IDs. They are these IDs. At this point, we can go to another database by using these IDs. So how do we do that? So first let us write this in a file. So I'm going to write this in a file. So I use the write command and say my gene ID is 2, comma, file. I have to give a file name. That is also my gene IDs dot txt. And then I don't need to do anything else. So I simply write this. And here I can see that it has written a file called mygeneids.txt. Let me click on that. And there are some NAs. And I have got this numbers here. So this is in a format which is not very convenient. So what I will do is I'll rewrite this file in a little more convenient way. I'll say n columns equal to 1 and let me see if it gives it a little more convenient. Yes, it is definitely more convenient. See, it has overwritten the file and this is in a single column. Now, there are some NAs here which I can sort of delete. I could have done it here also. So let me do this again so that we can do it in the script way itself. So my my gene ID is 2 NA indices. I write and I ask which my gene ID is 2 equal to NA. Sorry, I made a spelling mistake here. So it couldn't identify this Na. So there is another function in R called is.na. Is.na. So those are not available values, and this is the usage it is giving. So what I will do, I will indirectly do it in a different way. So my gene IDs 2 is NA equal to is dot NA my gene IDs 2. I think it is. So you can see the help file here, and the help file gives us uh, the arguments how we can give and how we can use the R function here. So you can see here that my gene ID is NA is a logical result. False, 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 like that it is coming because there is some number here. And then there will be some where it is true. So what do? We take my gene IDs to is underscore NA true is equal to which my gene IDs to is NA equal to true. Now it will work. So you can see here that the 13th element, the 14th element, like this, there are some element where NAs have been introduced. For example, my gene IDs 213. It's an N. Now we have to remove it. I don't like doing it manually, let's say. Okay? So let's do it through the computer. So my gene IDs to new, I call it, and then say my gene IDs to square bracket and give minus because I want to remove it, minus, and these numbers which I got from the which function, which will give me the places where this NA is there. So my gene IDs to T. Now I got my gene IDs to new. Supposedly, this NAs have gone now. Now again, let's write this write function, but we have given a different data object. So my gene IDs underscore new and press enter. So now I have got this file. which is not having NAs anymore. See, I'm scrolling. No more NAs. NAs are gone. The which could not identify the NA because which probably needs what is the, so there is a function special for NA which is is.NA. Like that is.null is also there enough. So this has given us these IDs. Now what we do is that we want to get, let us say, what are the pathways, what are the gene ontologies associated with all these genes. So the அப்புறம் கொடுக்குற சொல்லு. So this is string database. So this is one place from where we can get this data. And we click on multiple proteins. Actually, that's probably the best option you have given. miRNA is not a protein, no doubt. All the same, we will put it. So let string filter it out. So I have it here, all these IDs. These are Anthrax gene IDs. And I have said select all, copy, and then I enter it here. And there may be human, there may be mice, multiple IDs may be there, but I would select human for the time being and click on search. So there are some IDs for which the string found no proteins, probably because it's a miRNA. Then there are some IDs for which the gene has been found. So we have this ACSL4 that we have seen. We have seen this gene coming there. Then this one it has not found. The next one it has found, GPX4. And GPX4 has come again. So like this, there are these genes which have been found, and there are other IDs for which no proteins have been found. So like this, we can go through each page after page by clicking here next. Now we got the second page. So this is MAP4K2, vascular endothelial factor. Some of these are repetitions, no doubt, because we just gave the data as such. We can try to reduce the repetitions also. So how do we do that? We go back to R and we have this my gene IDs to new, which is having these repetitions. So my gene IDs to new equal to union my gene IDs to new, and write another time this same name of the object. This union function is a set theory function. It is not a biological function. It's a set theory function present in our base package. So we are using that. And what does it do? It does various different things. The set theory, you might have, I don't know when you know about it. Let's say you know little bit about it. Then the union function, what it does is it will remove the redundant, and because it is union of the same thing, so I'm going to get the same thing only, but the redundant IDs would have removed. So you can see here that from initial 658, after redundant removal, the IDs have now become 350. So let's write this function again. Enter, and there you go. Here we have the 350 IDs. Now let's again go to string and try to give this. So simply have to go back one more time. There it is. So I select all, delete, and now I go to R, and now I say select all, copy, and go to this string here, and again I select Homo sapiens and search. So yes, now we have the same thing, but the repetition is gone. Repetition is gone. VGFA has come only once. Repetition is gone. So this is good. And some IDs it has mapped, some IDs it has not mapped because they are not proteins. So string is about proteins. Pathways is about proteins. So this is one aspect of the data that we are looking at using string. So we can click on next here at the bottom. The next page opens and we can again see the different, the inflammasome protein has come, the apoptosis proteins have come, caspase, then again apoptosis proteins are there. So like this, this is the OGG protein, DNA repair enzyme, then fibroblast growth factor, keratin, and so on. Then there are some IDs for which the string has not found anything, which may be miRNA. Now, assuming that all are okay, we say continue. This is an assumption we are making because we are not going page after page. So by doing this, now we have found that the string has output a network of interactions between the proteins, which includes pathways and regular interactions, binding, and so on. It looks quite complicated, so and also quite dense. And we can see here that there is a central area where there is lot of connections between these proteins in which research is happening in diabetic retinopathy. And it makes sense because no doubt that because these proteins are having this high densely connected network, we are not surprised to find that much research is happening on this collection of proteins. Now, because it is too dense and it's also very complicated, so some of the basic statistics is here is that the network statistics is that the number of nodes is 170. So from 350, it has taken 170 on which the data is available in the string database. And the number of edges are between them, between the different nodes. And average node degree is 23, which is quite high. That is, on an average, each node is connected to other nodes, 23 neighbors. That's a lot of neighbors in the connection. So this is the basic network statistics for this network. Now we can see that this is a bit too dense. So usually we go to this setting and we would like to increase the stringency or confidence in the network. So instead of 0.4, we give a higher value. So let's say high confidence. So the more and more you increase the score, the more and more you can get the network with more clarity. So we click on update and there we see that the density is much reduced. Now we can see some spaces in between them. Okay. So in a pond, if you go and see that it is full of lotus densely growing, you cannot see any space between the plants, plant leaves, and water is not visible. You think there is water, probably feel there is water, you think there is water, because there is something different about the geography in that space. But you cannot see water, not a drop. It's so dense. And then you throw some stone or something, the leaves move, and then you can see the water. So in the same way here we can see that by increasing the confidence of the score, we get to see the network more with more clarity. Now we have the same VEGFA protein is there. This is FGF7. This is VEGFA. And by moving these marbles, so each of this node or a gene and protein, it is a marble in string. The same marble which we used to play when we were children. You can see inside. There are some things. Can you see? I hope it's visible. You can see when I move the mouse, it becomes a little brighter and the inner portion of the marble looks there is something is there. You can click on this marble and it gives us the information about the set gene. And that gene here happens to be vascular endothelial growth factor A, growth factor active in angiogenesis, vasculogenesis, and endothelial cell growth. Binds to FLT1 VEGF1 receptors. So the basic information about this gene is given in this little snapshot. In fact, even the crystal structure, not crystal structure, here it's the homology model of this protein is also given here. And the domain structure of this protein, as it has been identified through the PFAM and other algorithms, that is also given here. And this part has been homology modeled. So that's what is shown here about this. So then there are many other data we can get about homologs and about its relations with the pathways and other details as well. And you see, this is the UniProt link here. So I right click, open link in new tab, and it should open the UniProt link to this protein. So there you go. The UniProt site is opened. So this is a beautiful way of interconnecting between the databases in order to get information about the proteins that were being studied in a particular given disease. Isn't it amazing? From NCBI, we download the papers in a bunch, and then we take out the IDs through our scripts, and then these IDs we put in the string which maps it to the proteins, some of it, not all of it, because there are miRNA IDs also, which we will not get in string, we will get in some other database, And then we get it all integrated to these different databases, which gives full information about what is the gene being studied in the disease, what is its function, and that way we come to know the gene view of any disease. This is all possible because people have constructed databases like this using normalized IDs, and through our scripts and functions, we are able to get all this data together through analysis in a very smooth manner. The time I took to write the script and get the work done by the computer, how much it is, if you had counted it, it would have been less than 10 minutes. And the rest, we are spending time on reading about this protein, knowing what it is, seeing the network and all those things. So it is difficult to keep the computer busy, really difficult. So it has done its job, and now what we are doing is we are trying to gather information from different sources using this integrative approach. So we are going back to the screen. And now you can see that this is also linked to KEGG pathway. This is also linked to ensemble genome information. And the other data on the chromosomes. And then this RefSeq, which happens to be NCBI database. To that also they have given link. So I can right click and open the link for the KEGG here. So the KEGG pathway has come for the vascular endothelial growth factor. And we can see here that it is present in so many other pathways. So its importance is also with the microRNAs in cancer. So vascular endothelial growth factor is really very important, and there you see the diabetic retinopathy link has come. That's what we originally started with, and now we can see that this has been mapped to the disease part. Then there are drugs also for this. All these are drugs which are monoclonal antibodies that target the vascular endothelial growth factor, and this therapy is given in the indications where giving this therapy helps the patient to overcome from the disease. So it's a drug target, and as we scroll through, we can get all the more details, including the nucleotide sequence and protein sequence. All have come. And then from here, further analysis can be done depending upon the question one is asking. So I now click somewhere here so that it disappears. And this way each marble can be clicked and volumes of information can be obtained based on the genes and their functions. Now, I want to spend a little time on these connections between the marbles. What are these connections? So this is the meaning of network edges. So the line color indicates the type of interaction evidence. So they have different interaction sources, such as the text mining, experiments, about the interaction between the protein, or often the proteins which function in the same pathway or that are linked to multiple pathways, they have this tendency to get co-expressed. So both the genes will be co-expressed, and both the proteins will be made together, so that once this enzyme function is done, then the other enzyme function will also be done. That type of organization is there in our genome as well. Gene fusion, co-occurrence, and neighborhood. Neighborhood meaning nearby in the chromosome. So these are multiple evidences which are being used to have a neighborly connection between the different genes. So you can click here and the moment you click, then you can see that the different types of evidences they have mentioned here, and the combined score is 0.813. Neighborhood in the genome, not significant. Gene fusion, no. Co-occurrence across genomes, no. Co-expression, none. But putative homologs are co-expressed in other organisms. So that way it gets a modest score of 0.091. Then experimental and biochemical data, yes. And that's why it gets a very high score here, 0.6. So it's between 0 and 1. Okay, 1 is the highest. In addition, putative homologs were found interacting in other organisms. So that again puts a score of 0.36. Then association in curated databases, nothing. No significant evidence has been found. Then co-mentioned in PubMed abstracts, that is, authors, when they know that two proteins are working together, two or three, whatever, is their system of investigation, then they will keep mentioning them again and again. That is called co-occurrence. So that is non-significant here. So all the evidences are not very significant. So finally, a combined score of 0.8 has come, but our cutoff was 0.7. So because our cutoff was 0.7, 0.8 is more than 0.7. So that is why this connection has showed up. And that is the connection between these two proteins here. Now, in the same way, we can go through the other links also. And VGFA is a very important connection. So it is connected to even like this. This is hypoxia inducing factor. This is EGFR. And, and it can click on this hypoxia inducible factor 1 alpha, for which Nobel Prize was given for discovering this factor. And that is again connected to VEGFA. Can you see that? This is VEGFA and this is HIF1 alpha. So it's connected to VEGFA. You can move this around. You can little bit move this around like this, and then you can download this picture. So this is the display mode. You can click on export and then you can say download bitmap image or a high resolution bitmap. Any of these formats you can download, and that can be used for your study. Now, here we can see that for each node, they have given a table here between node 1 and node 2. What is the connection between the two and what are their scores? So pairwise, all these connections are given here. And then here we can see another button is here called analysis, which gives us the network statistics. And now you see when we had the medium confidence 0.4, then the average node degree was 23. The moment we increase the stringency and said, give me high confidence network, then you see the average node degree has immediately dropped by 1/4. It has now become 8.34. Not many are connected. And true, that is how we could see that the density of the network got reduced. And we are able to see more spaces in between the entities, the genes or proteins. And we can see here that the different gene ontology processes that come out very significant uh, in this corpus that we mined. But mind you, it was a subcorpus of miRNA. And we are looking at the genes in that miRNA. So this is not the full picture of diabetic retinopathy. This is a part of the regulatory process in the disease process of diabetic retinopathy. And we can see here that as we scroll down, the cake pathways are given. And we can see some pathways, they are coming, including a pathway called malaria. Now, what's malaria pathway doing in diabetic retinopathy? So, the genes which are involved in this pathway and the genes which are involved in the diabetic retinopathy, there are some genes which are common between the two. So, whenever there is a disease process, there are multiple genes involved in our system. Some are involved in defense mechanism because our body also has a system to protect itself. It wants to protect itself also. It will try its best. And then there are other processes which are going to destroy it, such as reactive oxygen intermediates and so on and so forth. And then in malaria, the infectious agent is involved directly in destroying some parts of our body. So, this type of linkage is not unusual to find. Then comes the next pathway, which is really, really very important, age-range pathway. It is on advanced glycosylation products in diabetic complications. And diabetic retinopathy is a diabetic complication. And that signaling pathway is highly involved. So what are these products? These products, so there is a system in our body to clear the modified amino acids, glycosylated amino acids, various different types of amino acids, which come from where? A lot of it comes from fried items, eating fried things. We like lot of fried things, French fries, fish fry, then so many fries, Pakoda, all these are fried items. Samosa, all these are fried items. If you keep eating those fried items, then this pathway is the one which has to remove the poor stock substances of modified proteins and amino acids and detoxify. If it doesn't, then it leads to complication, most notably inflammation, and that inflammation again creates insulin resistance, then it also causes a lot of damages to other organ systems in the body. Like this, one can go through all the data that is appearing in the string, and what they are giving here is the one which is high confidence from statistical point of view, so that the false discovery rate is extremely low. And you can click on more here, then you can get the entire table. And then as you scroll down, you can download each of these pathways in a tabular form. So let me download this get pathways, and here it is, save as Nextgenhelp. So it's a TSV file, and now I can go to Excel and click open, go to Nextgen, all files as before, and here it is, open, it's delimited. TSV means tab separated values. So tab delimited file, next, and finish. And this is the enriched cake pathway from the data we got. And here you can see that at each level, the age range has come, and this is the false discovery rate, which is very low. The matching IDs are given here, and these are the proteins that were found in the literature. So what people are really studying is they are studying the roles of these pathways in the role of the disease process. With this, we come to the end of today's session. I can take some questions.